Aerobic cellular respiration occurs when there is a presence of oxygen, but this will all start from glycolysis. Glycolysis takes place in the cytosol, and it's the metabolic pathway that converts glucose C6H12O6 into pyruvic acid CH3COCOOH product. The free energy released in this process is used to form two molecules of pyruvic ATP and ADH and water. After glycolysis is the preparatory stage, which is the pyruvic oxidation. Pyruvic oxidation is the conversion of the pyruvate into acetyl CoA by the enzyme complex pyruvate dehydrogenase complex. This is important as it is the step that connects glycolysis, which is our first step, and the Krebs cycle, which will occur after the pyruvic oxidation process. This process takes place in the mitochondrial matrix in eukaryotic cell and in cytoplasm to the prokaryotic cell, and it will produce CO2 and ADH and acetyl or CoA.
After pyruvate oxidation as well as another Krebs cycle will occur, Krebs cycle or the citric acid cycle. This is the main source of the energy of the cells and an important part of the aerobic respiration. The acetyl-CoA we made on the previous step will react on the first step of the 8-step sequence of the reactions that comprise the Krebs cycle. All of known takes place inside the mitochondria of the eukaryotic cell. This cycle produces 2 molecules of carbon, 3 molecules of NADPH, 1 molecule of FADH2, and 1 molecule of ATP or GTP. The last step of cellular respiration is the electron transport chain. This process is a series of 4 protein complexes that couple redox reactions, creating an electrochemical gradient that leads to the creation of ATP in the complete system named oxidative phosphorylation. It takes place in the mitochondria in both cellular respiration and photosynthesis. As I've said, in this process produces most of the energy as it makes 34 ATP molecules compared to the 2 ATP molecules made in glycolysis and Krebs cycle.
So let us discuss the anaerobic respiration. This takes place in some prokaryotic organism that lives in the environment without oxygen. This organism had an electron transport chain but do not use oxygen as a final electron acceptor at the end of the chain. Instead, they use other inorganic molecules as an electron acceptor. For example, sulfate-reducing marine bacteria use sulfate ion. First is the methanogens, a heterotrophic primitive archaebacteria that used carbon dioxide as the electron acceptor. Second is the sulfur bacteria. They do the same thing methanogens do, but the difference is they use SO4 as the electron acceptor.
So now we will be discussing about fermentation. It is used by animals such as ourselves when our body is not getting enough oxygen. This process does not require oxygen or any electron transport chain. In other words, it does not involve respiration. The first step of this process begins with glycolysis. This process occurs in the cytoplasm and does not require oxygen. Glycolysis oxidizes glucose to 2 molecules of pyruvate, which will be used in the next step. The next step depends on the type of fermentation that will occur. There are 2 types of fermentation: alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvate is converted to ethanol, or also known as ethyl alcohol, in 2 steps. The first step is the carboxylation, wherein it releases carbon dioxide from the pyruvate, which is converted to the 2-carbon compound acetaldehyde. In the second step, acetaldehyde is reduced by NADH to ethanol. This regenerates the supply of NAD+ needed for the continuation of glycolysis. In summary, the final products of alcohol fermentation are NAD+, ethanol, and carbon dioxide. Meanwhile, lactic acid fermentation is a one-step process and is most common in animal cells, specifically muscle cells, because they can regenerate NAD+ without carboxylation, making it a one-step process. They use an enzyme called lactate dehydrogenase to reduce pyruvate, which is a product of their first-step glycolysis by NADH. This reaction forms lactate or lactic acid as an end product and regenerates NAD+ with no release of carbon dioxide. This closes the metabolic cycle, which means glycolysis will only continue as long as there is glucose. In summary, the final products of lactic acid fermentations are NAD+ and lactic acid. The difference between aerobic and anaerobic is anaerobic fermentation produces much less energy than aerobic respiration, only 2 ATP per glucose processed as compared to 36 ATP produced by aerobic respiration per glucose molecule.